Good morning, everyone. Couple announcements to watch for on BSpace. So this week I'm going to be holding my office hours tomorrow instead of Friday. So I'll post that later today, the time of office hours tomorrow. Also next week I want to hold a couple of extra office hours because I know it's right before the exam. So watch for that as well. I'll post the announcement on BSpace later today or tomorrow for the extra office hours for next week. Today we're going to continue our discussion of the way that cells harvest energy from food. And we're going to talk about aerobic metabolism. This is the book chapter 9, pages 170 to 177. And I want to start off by talking about the citric acid cycle, and then we'll talk about oxidative phosphorylation, which is where most of the energy is actually utilized to make ATP. So remember that last time we talked about glycolysis, and you might remember that in glycolysis, what's happening is that we're starting with a 6-carbon sugar, glucose, and we're breaking it down to 2 3-carbon molecules of pyruvate. And that's where we stopped last time. And what we want to do today is pick up where we left off there and talk about what happens to pyruvate in the next steps of metabolism. So what we're going to see is that in the citric acid cycle, this cycle basically completes the energy yielding oxidation of pyruvate to carbon dioxide. In the process of those oxidation reactions, and remember it's a whole series of redox reactions, right? So the pyruvate molecules are being oxidized, and in the process we're reducing molecules of, remember from last time? Our old friend NAD+, and we're going to see another related coenzyme today, FAD. Okay, so both of those molecules are starting off in an oxidized state, and they get reduced in the process. And those molecules are actually the molecules that are most important for the production of ATP during oxidative phosphorylation. All right. So the first step is that glycolysis is going on in the cytosol, and so the first thing that has to happen to start the citric acid cycle is that pyruvate has to enter the mitochondrion, because it's inside of the mitochondrion where the citric acid cycle and oxidative phosphorylation are actually going on. And so what happens is that pyruvate is converted to acetyl-CoA. Okay, and this is an essential step that links the citric acid cycle to glycolysis. And I wanted to just show you, this is actually the chemical structure of coenzyme A. Your book doesn't show you this. And I don't know if you're like me. I was a chemistry major, so if you're like me, I always like to know the chemical structure of the molecules that are being referred to. I don't expect you to memorize this structure. Okay, but I just wanted to show it to you so that you know it's not just a blob in the book. And a couple of interesting things to note. So again, here's our friend adenosine, right? So we've got the adenine ring, glycosidic bond. Here's the ribose. It's got a phosphate on it over here on the 3' carbon, and then we have a diphosphate linkage to this long chain. And at the very end is a sulfhydryl, and it's this sulfhydryl that is actually the functional group that gets linked to pyruvate to make acetyl-CoA. All right, so here's one molecule of pyruvate, and it has to be actively transported across the mitochondrial membrane. Okay, so remember that active transport means that we have a transport protein that's in the membrane. It's specific for pyruvate, and if we're talking about active transport, that's an energy-requiring process, right? So it takes some energy to transport pyruvate across this membrane, and in the process, there's an oxidation reaction, so we lose a molecule of carbon dioxide, and there's actually a large multi-enzyme complex that is responsible for catalyzing each of these steps in this process. So the release of CO2, reduction of NAD+ to NADH, so here's where we're getting one molecule of NADH in the process, and coupling this acetyl group to coenzyme A to produce acetyl-CoA. And it's this molecule right here that can then actually enter the citric acid cycle, okay, as acetyl-CoA. And I wanted to just quickly remind all of us about the mitochondrion, because we talked about this a few lectures back, but it's now becoming very clear why the mitochondrion structure is set up the way that it is for metabolism. So just remember that, so these are organelles that are in most cells, and virtually all eukaryotic cells. They have a smooth outer membrane and a very highly enfolded inner membrane. So very importantly for this whole process, especially for oxidative phosphorylation, is the fact that the mitochondrion has a double membrane, right? A double membrane. And the inner membrane creates compartments that include the intermembrane space, so that's the space between the two membranes, and then inside of that is the mitochondrial matrix. Okay, so the citric acid cycle is actually going on inside the matrix of the mitochondrion. And that's what this is referring to here, is some of these metabolic steps. Respiration, this is the citric acid cycle. And very importantly, the cristae, which are these very highly enfolded, this part of the inner membrane, see how it folds in on itself. It creates a very large surface area inside the mitochondrion. This is really, really important for respiration, because inside of that membrane that forms the cristae is where all of the complexes that are responsible for oxidative phosphorylation are sitting. So the mitochondria are basically the little energy power plants of our cells, right? And so cells that have to generate a lot of energy, like muscle cells, will have a lot of these. And furthermore, each mitochondrion itself will have a lot of complexes inside the cristae that are generating ATP in the pathway that we're going to talk about today. Okay, so that's just a reminder of where all of this is going on. Okay, so citric acid cycle, other words for this, we talked about the tricarboxylic acid cycle, TCA cycle. It's also called the Krebs cycle in honor of the guy that actually discovered this pathway. All right, and so it's going on within the mitochondrial matrix, and basically it's oxidizing organic fuel that comes in the form of food derived from pyruvate and generates 1 molecule of ATP, 3 molecules of NADH, and 1 molecule of a related dinucleotide FADH, FADH2 per turn of the cycle. Okay, and then this is just again showing you the structure of these coenzymes. NADH we talked about last time, and new today is this related molecule FADH2, flavin adenine dinucleotide, which is shown over here. And again, you don't have to memorize those structures, but I do want you to know, you should be aware that they all have this interesting common feature of an adenosine molecule that's been decorated with other things that make up these coenzymes, and that they play an essential role in these redox reactions by being able to reversibly accept and donate electrons during these pathways. So this is an overview of the citric acid cycle. So pyruvate is coming in as a 3-carbon molecule, and it enters the mitochondrion here. It gets coupled to coenzyme A to form acetyl-CoA, and then it's this molecule that actually enters the cycle. And as we're going to see, this is actually it really is truly a cycle, because we start off with an enzymatic reaction here, and we go through 8 reactions, and we end up with a molecule here that is able to then re-enter the cycle. Okay, so it really is a cycle that's going around and around. Enzyme catalyzed, of course, right? And these are enzymes that are inside the mitochondrial matrix, but most likely they're associated with each other, because these are highly coupled processes. So as soon as one reaction occurs and the product is released, that product is then the substrate for the next enzyme, right? And so this pathway is just these enzymes are highly coupled to each other. Oh, right, and I just wanted to remind you that, of course, as we go through the cycle, we're talking here about 1 molecule of pyruvate, but we have to multiply everything by 2 if we want to calculate the yield we're getting from a glucose molecule. Yes? Sorry, I missed the second half. Why is the coenzyme A added, but? Well, it's a way of activating it for the next reaction, because it gets coupled to that sulfhydryl, right? And that's actually a bond that is more easily broken, okay? So it's just a way of providing a substrate for that enzyme that is activated already for the reaction that it has to carry out, okay? Yeah. Because, why is it multiplied by 2? Because each, we have 1 molecule of pyruvate is shown here, but from each glucose molecule, we have 2 molecules of pyruvate that are made. So if we want to know the total yield for 1 molecule of glucose, we have to multiply this by 2, right? Okay, so the citric acid cycle has 8 steps, and as I mentioned, each of these is catalyzed by a specific enzyme. Okay, and so the acetyl group of acetyl-CoA joins the cycle by combining with a molecule of oxaloacetate to form citrate, and then in the next 7 steps of the cycle, the enzymes break down this citrate back to a molecule of oxaloacetate, and that's what makes this a cycle. And in the process of doing that, molecules of NADH and FADH2 are produced, and these are basically then the way that the cell is essentially harvesting electrons through these redox reactions, and then as we'll see, it's going to use those molecules to drive the production of ATP during oxidative phosphorylation. Okay, so this is how we get from food to an electron transport chain to ultimately a very cool little machine that is actually sitting in the mitochondrial membrane, and as we're going to see, we'll see a little movie today, it actually really is a little motor that is literally cranking out ATP. Okay? All right, so let's just walk through the steps of the pathway. And again, I don't expect you to memorize all of the structures of the intermediates in the pathway. What I do want you to know is things like, there are 8 steps in the pathway. What do we need to get into the pathway? We need pyruvate, but we need it not as pyruvate, right? It has to get coupled to coenzyme A to make acetyl-CoA, so you know that. And then you should know the overall yield of the pathway. Okay, how is energy actually harvested during this cycle? All right, so as we mentioned, so in the first step of the pathway, we have acetyl-CoA coming in, and immediately that CoA is released, and this reaction that's catalyzed here couples that group to an oxaloacetate molecule. So this is coming in from the end of the cycle on the other end. As we'll see, we're going to end up with this. So these guys get coupled, and we end up with a molecule of citrate, and it is a tricarboxylic acid, right? So there's the TCA, the tricarboxylic acid. So we start off with this molecule of citrate, citric acid. And as we go through this, you'll see that your book has this figure, and they basically are showing us the 2 carbons of the entering acetyl group from the pyruvate shown in red here, and the other carbons that are coming from the oxaloacetate molecule in blue. And so you can at least follow for partway through the cycle what's happening to those 2 carbons that enter the cycle. Okay. All right, and so in the next step, citrate is converted to its isomer isocitrate by removing 1 water molecule and adding another. Okay, so that's again an enzyme catalyzed reaction responsible for that. And then in the third step, right here, we have isocitrate being converted to alpha-ketoglutarate. And in the process, we're releasing another molecule of carbon dioxide with a carbon from sort of old carbon, right, a carbon that has come through the cycle through oxaloacetate. And in the process, we also reduce 1 molecule of NAD+ to NADH. So here's the first step of the cycle that actually leads to transfer of electrons that are being harvested for later, right, through the reduction of NAD+ to NADH. Okay, and then in the fourth step, right here, alpha-ketoglutarate is oxidized to succinyl-CoA. So again, we have formation of one of these sort of high-energy bonds here with a sulfhydryl group that is coupled to this succinate through coenzyme A. And we also get a reduction of another molecule of NAD+ to NADH and another molecule of CO2 that gets released in that pathway, in that process. Okay, and then in step 5, right here, this high-energy bond is broken. We get release of a succinate molecule. We recycle that coenzyme A that can go back and do this earlier reaction again. And we also have an inorganic phosphate that comes in and is used, again, enzymatically to phosphorylate a molecule of GDP to GTP. Okay, and that's very highly coupled to a substrate-level phosphorylation event that converts ADP to ATP. Okay, so remember that GDP, it's sort of analogous to ADP, right? Except it's a guanosine dinucleotide instead of an adenosine diphosphate, guanosine diphosphate instead of an adenosine diphosphate. But so this is a reaction that is utilized directly to phosphorylate ATP over here. So this is the one step in the citric acid cycle where we're actually generating a molecule of ATP directly through substrate-level phosphorylation. Okay, and then we go to the next step, and here we see this molecule of FAD that comes in. It actually gets reduced to FADH2 during conversion of succinate to fumarate, so another enzyme-catalyzed redox reaction there. And then the fumarate molecule is then hydrolyzed with water to produce malic acid or malate. Over here, we're almost there. One more step, and that's this reaction right here, step 8, that again leads to a reduction of NAD+ to NADH, and in the process we convert malic acid to oxaloacetate, so we're back where we started, right? And so this molecule can then re-enter the cycle when another molecule of acetyl-CoA comes in to make citrate, and we go back around the cycle. So the cycle is going around and around. We're generating these, we have these redox reactions going, and substrate-level phosphorylation making 1 ATP, but mostly we're making NADH and FADH2 that we're going to use later in oxidative phosphorylation. I thought I saw a hand. Somebody want to ask a question? No, you changed your mind. Okay. Okay. So if we go through just, what's the total tally of what's being produced for each acetyl group that enters the cycle, right? So we have 3 NAD+ molecules that are being reduced to NADH, and those are in steps 3, 4, and 8. And then we have 1 molecule of FAD that's being reduced to FADH2 that's being produced in step 6. And then we have 1 substrate-level phosphorylation going on in step 5 that makes a molecule of ATP. Right? And so by far, most of the ATP that is generated during overall cellular respiration is actually not being made during glycolysis or during the citric acid cycle. Right? There we're only making 4 molecules of ATP. Most of it is made during oxidative phosphorylation, where these molecules of NADH and FADH2 that have been made here and earlier are used to transport electrons in a process that involves an electron transport chain of enzymes. Okay, and so let's look at that a little bit. So during oxidative phosphorylation, we have a process of chemiosmosis. that couples electron transport to ATP synthesis. So remember back when we talked about the structure of the cell and the fact that when we have cell membranes, one of the things that membranes can do is they can be only sort of partially permeable, or they certainly have ways of regulating the permeability of molecules across the cell membrane, and not only in a chemical sense, but also in an electrical sense. So somebody asked me last week in office hour, maybe it was earlier this week in office hours, about a chemical gradient versus a voltage gradient across membranes. We can have both, right? So if we have a chemical gradient, that means that we have a higher concentration of a molecule on one side of a membrane than the other, and if that molecule is an ion, then that can also set up an electrical gradient across the membrane, right? A voltage gradient. And so basically that when we set up that kind of situation, we have a way to then drive the transport of molecules across the membrane by allowing them to then go across move sort of down their concentration gradient, or move in a direction that's favorable based on the electrical gradient across the membrane. So what we're going to see in this process of chemiosmosis is that we use energy to create an electrical and chemical gradient across the membrane, in this case the inner membrane of the mitochondrion, and we're going to actually use that gradient to drive the process of ATP synthesis through this very interesting motor called ATP synthase. Right, so following glycolysis and the citric acid cycle, as I mentioned, we're getting some ATP made. So we have 4 molecules of ATP that are made, net gain, that are made by substrate level phosphorylation in those two processes. But by far, most of the energy that is harvested by cells through this whole long series of enzyme catalyzed reactions is through the reduction of NAD and FAD to NADH and FADH2. Right, so these account for most of the energy that's extracted from foods in the form of these molecules. And so these are 2 electron carriers, and what they do is they donate electrons to the electron transport chain that we're going to talk about next, which powers ATP synthesis using oxidative phosphorylation. Okay, now did I see another hand? No. Okay. Food is the initial source of the energy that's coming in, right? So that's where we're getting those molecules from. Okay, so the pathway of electron transport. So as I said, so the electron transport chain is located in the cristae of the mitochondrion. Remember that that's this really beautifully enfolded membranous structure that we see inside of the mitochondrion. And so as we talk about this pathway, just imagine that you've got these enzyme complexes that are seated in that inner membrane, and we don't just have one complex, right? We typically have per mitochondrion, we might have hundreds or thousands of these. So these little factories that are embedded within that inner membrane, and the fact that it's highly enfolded is great for this purpose because it creates a very large surface area. So we can just have more of these little factories embedded, and so it makes the mitochondrion that much more efficient at doing these reactions. So as we're going to see, most of the electron transport chain's components are proteins. And furthermore, they're not just one polypeptide, but typically these are large multi-protein complexes that interact together to create this literal sort of chain of electron transport reactions that are occurring in the membrane. So they're not just sort of isolated reactions, they're actually physically associated with each other. And we actually know, again, you might wonder how do we know all this, and a lot of it we know from X-ray crystallography work that's been done to actually look at molecular structures of these enzyme complexes and figure out how they work. And then there's also been a lot of nice work, so back, looking at the pathway that we just talked about, the citric acid cycle, there we know a lot about the transfer of carbons from one molecule to the next in that pathway by doing experiments with carbon labeling, right? So you can actually start with a labeled, let's say a C13 labeled pyruvate molecule, and just basically observe where those labeled carbon molecules end up at each step of the pathway. And again, this is where enzyme inhibitors or enzyme mutants come in experimentally, because we can use inhibitors to block enzymes in the pathway specifically and look at what compounds build up when we block a particular enzyme. So what product is being made that now can't be consumed in the next step of the pathway when we have a mutated form of one of these enzymes or an inhibited form of one of these enzymes. So that's just a little bit about how experimentally we know all of these kinds of details. Okay, so the carriers alternate reduced and oxidized states as they accept and donate electrons. So this is where lots of redox is going on. And electrons are dropping in free energy as they go down the chain. And eventually, they're actually passed off to oxygen to form water. That's the very last step of this electron transport chain, is the production of water. But as we saw last time, the cell doesn't want to let that reaction happen quickly or just happen immediately, because a lot of energy would be released and it would be very hard to harvest it. Plus, it would probably be pretty destructive for the cell, right? So this is a really beautiful way of coupling a whole series of reactions where relatively smaller amounts of energy are being harvested at each step. And in the process, we're storing up molecules that can then be used to generate ATP at the end. Okay, so let's take a look at this electron transport chain. And, the book doesn't have a really great picture of this. If anybody's really interested, there's lots of nice information that's really easily available on the internet if you do searches for these individual, like cytochromes, for example, that are part of this pathway, crystallographic structures and lots of cool, details about how these work. And it's sort of more depth than we can go into in Bio 1A, and you'll learn a lot more about this if you take biochemistry. But I want you to know some of the overall features of this. And so what your book shows is it shows a cartoon of the 4 major multi-enzyme complexes that are part of this electron transport chain. So these are shown as complex 1, complex 2, complex 3, and complex 4. Okay, and then there's lots of other stuff in there that we'll talk about in a moment. And it's showing you this cartoon drawn in this diagonal way because they're showing it to you as drawn with free energy over here relative to oxygen. And over here, sort of, I guess you could sort of consider this a reaction coordinate effectively, right? Because the pathway is sort of, electrons are flowing in this direction in the pathway. And so we start off with electrons entering in a fairly high energy state. And as each of these successive redox